Hi everyone, and welcome to Chapter 4, which focuses on cell structure and function. And yes, the answer is always the mitochondria is the powerhouse of the cell. Now, when we talk about cells, I first want to point out that they are ridiculously small. So this graph kind of shows you how small in comparison to things such as humans or insects, for instance. And you'll notice when you see this representation here, so for instance, between 10 to 100 micrometers, that's what that symbol stands for, micrometers, and that's usually 10 to the minus 6 if you were to write out what a micrometer is in comparison to a meter, where you then find Sorry, I should point out that we're not a meter, we are above 1 meter, but I'm just talking about the units there. Now, you may ask yourself, well, why is something so critical, so important to all of life, so tiny? Well, it's actually better to have a whole bunch of small cells making up your body instead of one really big one. And the reason that is that cells exchange materials based on their plasma membrane, which we're going to go more into in the next chapter. But when they exchange materials based on their membrane, they need to have a large surface area, meaning more surface area in order to exchange materials. So by having many small cells put together, you end up having a larger surface area than if you were to have one really big cell. And sometimes the way I have you kind of visualize it, one, you could use the cubes that are on the figures here. So one really big cube versus a whole bunch of small ones, where you then get all of that inner area as surface area. Or another example is to look at your own hand, right? Look at your hand and feel around each of your fingers all of that surface area, okay? All of that extra surface you have by having 5 fingers. Then next, squeeze your hand together so that all of your fingers are pushed up against each other. Well, now you have a whole lot less surface that you can feel, right? You no longer have the individual areas in between each of those fingers. Okay, so many cells put together have larger surface area than just one, and that's better for exchanging materials. Now, there are 4 things that all cells have that you should know. The 4 things they all have is a plasma membrane, cytoplasm, DNA, and ribosomes. Okay, so that's plasma membrane, cytoplasm, DNA, and ribosomes. Now, because cells are so tiny, you can't just look at a person and say, "Hey, I see your cells." No, you need special equipment. So in order to study cells, we use multiple different types of microscopes. And remember, we mentioned that in a previous lesson, that microscopes enhance our observation abilities. And you can see here on this slide how they differ from each other. Now, you don't need to memorize all the different types of electron microscopy and light microscopy and all of that. What I do want you to know is that the compound light microscope is the kind of microscope that you use in regular classes. So for instance, in a high school biology class, if you ever look through a microscope, that was a compound light microscope, and you look through it using your eyes to, look into eyepieces or oculars. That kind of microscope can have a maximum magnification of around 1000x. Okay, so I want you to know compound light microscope, kind that you've used before in, for instance, classrooms, and that the maximum magnification is 1000x. The reason that the max is 1000x is that those eyepieces that you look into have 10x magnification, and then the objectives that you turn at the bottom of the nose piece, the highest one is 100x. So 10 times 100 equals 1000. Electron microscopes are then more powerful. Okay, so electron microscopes allow you to see a maximum magnification of 1,000,000 times the image. Okay, so it's much, much more powerful. And in this one, you wouldn't be looking into eyepieces, you would have a projection of the image, and a lot of times you then view it on a computer screen. Same thing with a confocal microscope. That's another very powerful type of microscope, and that one you would not look through your eyes, but again, would end up looking on a computer. Okay, so these are just some examples of different microscopes. Now, when we talk about cells, one of the big concepts that we've mentioned already is you have to know the difference between prokaryotes and eukaryotes. You are made up of a whole bunch of eukaryotic cells, whereas something like bacteria is a prokaryote. And the trick that my student always used to remember the difference is think pro, no. Pro rhymes with no, so prokaryotes have no nucleus. Okay, so they do not have a membrane-bound nucleus. And when we talk about the nucleus, what we mean is in eukaryotes, the nucleus is the center of that cell, and it houses the DNA and the genetic material of that cell. It's basically like a command center of that cell. And we'll go more into detail in later sections, but I just want you to really know what I mean when I say no nucleus. Also, prokaryotic cells tend to be smaller and simpler than eukaryotes. So if you think of a bacteria versus a person's cell, bacteria is usually going to have those smaller, simpler kind of functions to their cells. Now, when we talk about prokaryotes, there are two domains you have to remember: bacteria and archaea. And bacteria, you've all heard of those guys, but you may not have heard of archaea. What you have to know about archaea is not only are they prokaryotic, but they're distinguished by the fact that they live in extreme environments. So if you found life in, let's say, a volcano, that would usually be an archaea cell. And they're very valuable to us for that reason, because we can get things like cool enzymes from them. The other thing I want you to know is when we talk about prokaryotes, we usually refer to their shapes a lot, and there are three basic shapes that they can have. They can have round, like you see here, and that is coccus shaped. They can have spiral shapes, which we sometimes call either spirillum or spirochete shapes, depending on whether they're very rigid or they're really fluctuating around. And they can be bacillus, and bacillus means rod. Okay, so I want you to be comfortable with, if for instance I ask you, what would you call a rod-shaped bacteria? I can give you bacillus, spirochete, coccus. You should know which one it would be. Or I could say, what does it mean if a bacterium or a prokaryote is bacillus? You should know that that means it is rod-shaped. So make sure you're very comfortable with those differences. Okay, here is a quick recap notes slide, just in case you missed some of what I was saying. I wrote it out here for you to pause and jot down some notes. When we talk about cells, or specifically prokaryotic cells, you should also know what the cell envelope is made of. And that's on the outside of cells because, as you know, envelopes package things, right? So cell envelope is made of three main things. It's a plasma membrane, which all cells have. Even your cells have a plasma membrane. And plasma membrane is so important that it gets its own chapter, which we'll discuss after this lesson. And the reason why the plasma membrane is so important is it has the important function of regulating what comes into and what exits the cell. Okay, so it regulates entrance and exit of substances. Okay, make sure you write that down for the function. In addition to the plasma membrane, prokaryotes have what we do not have on our cells, which is an extra cell wall around them, and that helps maintain their shape. Above that cell wall, some bacteria or some prokaryotes also have what's called a glycocalyx. And the glycocalyx is basically an extra shell, which a lot of times we will call either a capsule or a slime layer. And they left out the E in this figure, but it should be a slime layer. So that outer membrane in this picture here, you can write glycocalyx for that. That's what they're representing. It's either a capsule if it's tight around the cell, or a slime layer if it's loose around the cell. And either way, it's important because it's a nice protective capsule. And it helps those prokaryotes stick to things or attach to things as well. So make sure you're comfortable with what the plasma membrane is, cell wall is, and what the glycocalyx is. In addition to having that cell wall around them, some prokaryotes have some extra appendages on their outside, so outside of their cell wall, outside of their membranes. One of them is the flagella. So if you've ever seen a picture of E. coli, a lot of times they look like this guy here, and the flagella is their little whip propeller. Okay, they can have one or they can have many all over them. So the flagella is for movement. Make sure you know the function of each of these appendages we mentioned. So a flagella is this whip-like appendage that propels the cell for movement. Some cells also have fimbriae, which in this microscope picture, you see all of these little extensions sticking out, these little hair-like structures here. Those fimbriae are very important for prokaryotes because those allow them to anchor or grab a hold of surfaces, sometimes even other cells. So for instance, bacteria sticking through the lining in your throat or the lining of your nose, they're able to do that better if they have fimbriae, because fimbriae are for attachment. The last one here is conjugation pili. So this, you have a conjugation pilus here. Pili is plural. It's basically like this little tunnel that extends from one bacteria, grabs a hold of another bacteria, and allows them to pass DNA from one cell to another, because it will occur during conjugation, which is a scientific way of saying bacteria sex. If you think of conjugal visits, that can help you think of what conjugation means for bacteria. So please remember that a conjugation pili is how bacteria pass DNA from one cell to another during sexual reproduction. This figure here kind of helps summarize some of the structures that you might see on a prokaryote, including some of the ones we just mentioned. So we mentioned flagellum, that's the propelling movement. Glycocalyx is the outer protective shell. Cell wall helps with the shape of the cell. And plasma membrane is letting things in and out. And you also got to see these guys as well already, fimbriae. So just make sure you're comfortable with the functions of any of the ones that we've already discussed. In addition to prokaryotes, there are eukaryotes, and eukaryotes are important. Why? Because that's our cells. Okay, we are eukaryotic cells. And when it comes to eukaryotes, the big thing to remember is how they differ from prokaryotes is our cells have internal membranes. They have a nucleus. Now, what's interesting about eukaryotic cells like ours, remember in the first lesson when I asked you what was the first form of life? Well, we said microbes. And by microbes, we meant prokaryotes, right? Such as cyanobacteria. Now, what's really interesting is that the origin of eukaryotic cells, such as us, is that we came from prokaryotic cells. It appears that way back in the day, a prokaryotic cell engulfed, meaning basically ate up, and put a big old envelope around and chewed up other individual prokaryotic cells. And that resulted in one cell with multiple organelles and compartments, because think about it. If a prokaryotic engulfs, which you see, let me get the laser pointer, which you see here, it takes in other smaller bacteria. Well, each of those smaller bacteria, they have membranes around them, right? So now this prokaryote that started out without having any internal membranes now has engulfed internal membranes and eventually over time developed into what we now know as eukaryotes, which include animal and plant cells. Now, the way to remember that the endosymbiotic theory means that eukaryotes came from prokaryotes that engulfed each other, meaning prokaryotes that ate each other. Well, the easy way to remember that is ask yourself, what does endo mean? Well, exo means outside, endo means inside. So endosymbiotic means that prokaryotes took inside other prokaryotes, and ultimately we eventually got eukaryotes. So make sure you know what this term here means. Here's another recap note slide, just so that you can jot down the theory information in case I said anything too quickly. Now, with eukaryotic cells, there are a few domains that it includes. It includes animals such as us, plants, fungi, and protists. Now, I want you to circle, star, highlight the fact that fungi are eukaryotes. Because, well, first of all, what are fungi? Fungus is yeast or mold, right? Yeast and mold are fungi. Now, what does it tell you that fungi are eukaryotes? Well, it tells you that they have a nucleus, that they are more like us than they are like bacteria, because bacteria are prokaryotes. They don't have those internal membranes. The reason why I emphasize this so much is that I've heard students and even people outside of school constantly refer to fungi as bacteria. They'll say mold, bacteria. They'll say yeast, bacteria. No, 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 no. Two very different things. Mold and yeast are more like you than they are like bacteria. And that's why we use yeast for so many different experiments in scientific research. So, the important aspect of listing out the domains here is that I can ask you on a question, right? I could be like, okay, so which of the following does not have a nucleus? And I could list out bacteria or even specify a famous bacteria like E. coli. You know, E. coli is a bacteria. I could say E. coli, yeast, or human cell. Well, yeast is a fungus, so it has a nucleus. Human cells are eukaryotes. They have a nucleus, right? Because they're animal cells. The only one of those that does not have a nucleus would be the bacteria that I list. So specifically E. coli. Okay? Now, in addition to having a nucleus, eukaryotic cells contain other things as well. They contain specialized compartments, meaning membrane bound. So inside of the one cell, there's multiple little membrane bound compartments, which we call organelles. They have the plasma membrane, which we mentioned in the previous slides for prokaryotes as well. So we also have a plasma membrane that serves two functions for us. It's for material exchange and it's for protection as well, because remember, we don't have a cell wall beyond that. Okay, so our plasma membrane is for material exchange and protection, so getting things inside and out of the cell and protecting us. We also have a cytoskeleton, which is basically a whole bunch of protein fibers that do what our skeleton does in our body. It maintains the shape of the cell. So the cytoskeleton maintains the shape of the cell, and it also helps organelles move around a little bit. The last point that I make here is that plant cells and fungi do contain a cell wall, even though our cells do not. So some eukaryotes will actually have a cell wall, but it's slightly different from bacteria cell wall. It contains much stronger, different carbohydrates in it. Now, this slide here is to help you visualize some of the differences that we've been talking about between plants and animals, and they highlight in green versus red some of the differences that you'll encounter as we also go through the various organelles and structures that cells can have. Now, when we talk about eukaryotic cells, we said that they have internal membrane-bound structures, which we call organelles. And there are two different classes of these organelles. There's what we call the endomembrane system, which will communicate with each other via channels or vesicles. And vesicles, what that basically means is these little compartments or little sacs that the cell will produce to put things inside of. Now, the endomembrane system includes things like the nuclear membrane, things like the endoplasmic reticulum, and the Golgi apparatus. And we'll talk about these in more detail in a minute. Now, when we talk about these different structures, so again, endomembrane system being nucleus, endoplasmic reticulum, Golgi apparatus, and even the vesicles that you'll see produced from these structures. There's also energy-related organelles. And the energy-related organelles are the organelles that help that cell produce energy. And the two big ones are mitochondria, which both animals and plants have, and then chloroplasts in plant cells. Like I said, in the following slides, we're going to end up going through each of these different structures in more detail. Here's another recap slide so you can pause and jot down the notes just in case you were unsure of the spelling of any of the terms in the previous slide. Now we're going to go through some of the organelles we mentioned, what they actually mean. The first one is the nucleus, and you can think of that as the brain or the command center of the cell, because that is the organelle that will contain the genetic material of that cell. And in eukaryotes, this genetic material is in the form of chromatin. And what chromatin is basically, think about it. If you have DNA, a DNA strand is ridiculously long. Like if you unraveled it, it would be around like 6 feet long. And so how are you going to fit that in a tiny microscopic cell? Well, what the cell does is it takes that DNA and it wraps it around proteins so that now it's a nice, compact, squished up ball of DNA that can fit inside the nucleus. And there'll also be some RNA mixed in as well to help maintain that shape. Okay, so the DNA and the genetic material of that cell is found in the nucleus. There are also what we call ribosomes, and whenever you hear ribosomes, I want you to think protein makers. Ribosomes, their function is to synthesize proteins. They do this via what we call translation, turning an RNA code into protein. And you'll find ribosomes either on the endoplasmic reticulum, specifically the rough ER, so that's why it's rough, because it has all these little ribosomes stuck to the outside, or they'll be free-floating in cytoplasm. And sometimes if you see them in groups just free-floating in the cytoplasm, we call them polyribosomes. But I really want to emphasize the fact that when you hear ribosome, think making protein. Now, earlier we mentioned that there is the endomembrane system. What the endomembrane system is the compartmentalization of the cell, meaning picture endomembrane means inside membranes. So all of those membranes helping to separate the different structures in the cell. So that includes the nuclear envelope, the endoplasmic reticulum, the Golgi, and the vesicles. Now we've kind of already mentioned what the nucleus does. So the nuclear envelope helps hold in all of that DNA and genetic material. Next, we're going to talk about the endoplasmic reticulum. Now, some people think of the ER, or the endoplasmic reticulum, as the roadways of the cell. And that's because there's basically a whole lot of transport going on in these roadways. There's some protein transport and some lipid transport. Now, we've already mentioned the rough ER, and I said that it was rough because it has all of those ribosomes stuck to the outside of it, right? So it's lined with a whole bunch of ribosomes that make it look rough, and you could think R, rough ribosome. Now, as soon as you hear that the rough ER has a whole bunch of ribosomes, what does that tell you its function is? The rough ER can produce and modify proteins. Okay, so when you think of the rough ER, think of ribosomes and think of protein making and transport. And it also modifies the proteins as well, meaning it can add extra, extra element groups to them. So for instance, it might phosphorylate them, stick on a different group, okay? There's also the smooth ER, and when you think smooth, well, it's smooth because it does not have ribosomes on it. And when you think smooth, I want you to think waxy, oily, right? Oily smooth. Well, that's because the smooth ER can produce lipids, so that will help you remember. If you think smooth, even though they're smooth because they don't have ribosomes, when you hear smooth, if you think oily, waxy, it'll remind you that the function of the smooth ER is to produce lipids, which can include fat molecules and hormones. And then what they do, the reason why it's thought of as roads or streets, is they package up these molecules. So they form vesicles, meaning little round sacs, or picture a round bag. So they're bagging up these lipids that they just produce, and they'll transport them in vesicles to other parts of the cell where they are needed. Okay, so ER, or the endoplasmic reticulum, helps produce molecules and also helps package and ship them out. Now, the big packaging plant, however, is not the endoplasmic reticulum. The big packaging plant is actually the Golgi apparatus. And what's important about the Golgi apparatus is it will take in those vesicles that came from the endoplasmic reticulum. So if you look at the figure on this slide, you have the nucleus here. This is the rough ER because you see all of those speckles of ribosomes. Then you have the smooth ER, and inside the smooth ER are all those lipids that it's been producing and modifying. Now notice from the endoplasmic reticulum, there'll be these little transport vesicles that then enter the Golgi. This big structure here is the Golgi apparatus. So vesicles containing the produced lipids or the produced proteins from the endoplasmic reticulum then enter the Golgi apparatus, and in the Golgi apparatus, they then will modify those proteins and lipids, so they will change their structure a bit. They might add on certain molecules to them, and then they package them in new vesicles to then ship completely out of the cell or ship to other parts of the cell if needed. Now, if they are shipping a vesicle containing these modified proteins and lipids completely outside of the cell, we call that secretion. The cell has secreted those substances. And the way that they do that is the vesicle, you'll see in this picture, it merges with the cell's membrane and then spits out its contents. Okay? So remember that the ER was producing the proteins and the lipids and putting them in a vesicle to ship to the Golgi, which then modified them further and shipped them out of the cell or to a different part of the cell. Now, the Golgi will produce some very important vesicles that are not just, modified lipid or protein to move elsewhere, some of the Golgi vesicles produced are what we call lysosomes. And this happens in animal cells, not in plant cells. And what these lysosomes are is a vesicle or a storage sac full of very powerful digestive enzymes. And that'll be very important in the cell because the cell will then use lysosomes to help digest its materials that it needs to break down. In fact, it will even use the lysosomes to destroy any damaged organelles or damaged proteins that might cause problems within the cell. So it ends up using them as a basically recycling plant or a garbage plant. And even the lysosomes, scary enough, they're even involved in programmed cell death. So for instance, sometimes cells that will get infected, they basically know that they are a problem for the overall well-being of that cell or organism. So they will have programmed cell death where the lysosome will engulf and break down that cell, okay? Or release the digestive enzymes in it to destroy the cell. Now, if you think of the lysosomes, I want you to think of them as a big ball of digestive enzymes. They help break things down in the cell. This slide here is just to give you a summary of the endomembrane system that we talked about. So again, started off with the nucleus, which is the genetic material of the cell. You then have the rough ER coated in ribosomes for making proteins and modifying them. You then have the smooth ER, which is for smooth lipids, making fats and oils and hormones, and then putting them in transport vesicles to enter the Golgi. So notice the rough ER gave transport vesicles with proteins, the smooth ER gave transport vesicles with lipids. In the Golgi, the proteins and lipids are then modified, packaged into new vesicles, and may even be secreted out of the cell. And included amongst the vesicles leaving the Golgi, that may include lysosomes, which are the digestive enzyme organelles of the cell. Okay, make sure you're comfortable with each of the structures and their function. And if you have any questions or concerns, just send me a message and remind, and I will further clarify or give you some practice problems. Now, sometimes you encounter other vesicles or vacuoles in cells. One type is the peroxisomes, which are very similar to lysosomes, except they're specific to having hydrogen peroxide in them. Okay, so the lysosomes will have a whole bunch of different digestive enzymes, whereas the peroxisomes are specific to having hydrogen peroxide, which can be very toxic to cells. So it can be useful in breaking down things such as lipids, and can be valuable to helping the cells destroy things when it needs to. But at the same time, the rest of the cell has to be able to protect itself. So sometimes they will have what's called catalase, just in case any of that hydrogen peroxide gets out of the peroxisome. The cell will be safe because through catalase, it's able to break down that hydrogen peroxide into water and oxygen, which is safe for the cell. In plant cells, there's also another interesting vesicle or vacuole called the central vacuole. So whenever you see plant cell pictures, you'll see this huge empty gap, it looks like, in the center of the cell in pictures. So it's this big gap over here. That takes up a huge amount of the cell, and it's important because it helps maintain turgor of plant cells, which is the rigidity. That rigid structure of plant cells that you see is maintained by the central vacuole. And it basically functions a lot like lysosomes in animal cells. So it can have a lot of digestive enzymes and helps to break down old or defective organelles within the cell. It also serves as a storage container for water so that the plant cells will not have their cells constantly bursting from taking in too much water, because as you know, plants constantly want to take in water, right? So the vacuole serves as storage of water and also as storage of digestive enzymes to help break down what the cells need. Now, in addition to the endomembrane system, which included those vesicles that we were just talking about, even the central vacuole and peroxisomes, we also have the energy-related organelles. And like I said, the energy-related organelles are mitochondria, and in plants, it includes chloroplasts. Now, when you think of chloroplasts, again, think of these as being in plant cells, not animal cells. They are a double membrane enclosed organelle, and they use what's called chlorophyll to help capture sunlight. And the chlorophyll you find in what we call thylakoids. So when you see these little structures here that look kind of like a Mento, these little structures here are thylakoids stacked on top of each other. And those thylakoids have chlorophyll, which helps capture sunlight. And that's critical in things like plants, algae, and even certain bacteria that use photosynthesis. Okay, so chlorophyll you'll only find in things like plants, algae, bacteria, and the function is for photosynthesis, for turning sunlight into usable energy in the cell. Now, when you think of these, again, keep in mind you're thinking of cells that can produce basically their own food. They're producing food, they're producing energy for themselves. Okay, energy-related organelles. There's also the mitochondria, which you always hear called the powerhouse of cells, and powerhouse because it is the big energy maker of the cells. What they do is they carry out cellular respiration, which if you visualize the formula for that, you'll see in later chapters, it's actually the opposite of photosynthesis. So in photosynthesis, the plant cells or the cyanobacteria, they're making glucose, they're making sugar molecules, basically their food, and also energy. In the case of mitochondria and respiration, what's happening is glucose is being broken down. So instead of making glucose, they're breaking it down to get more energy. Now, the way that you see this happen is they will make carbon dioxide, water, and energy from carbohydrates, specifically glucose, using also oxygen. Now, when we talk about mitochondria, you'll see them as organelles that have two membranes. They have an inner and an outer membrane, and that will include this semi-fluid matrix with DNA and ribosomes in it. And so as soon as you see that they're capable of making proteins, right? Whenever you see the word ribosomes. And so mitochondrial DNA and proteins, they're basically encoding for and producing proteins that are involved in what's called electron transport. And also, sorry for the big scary term, you don't have to memorize it, also oxidative phosphorylation. And that's a fancy way of saying making ATP. Okay, that's involved in making ATP and energy. So you don't have to memorize those terms, just know that the DNA and ribosomes that you find in mitochondria, sometimes it's called mitochondrial DNA, they are used for making proteins involved in electron transport and ATP synthesis. Okay, that's why they're so important. Now we mentioned that there's also a cytoskeleton in cells, kind of like your own skeleton. It'll be made up of a whole bunch of protein fibers kind of stuck together and having motility. And these are very important because they have two main functions. They're involved in maintaining the shape of the cells, and also they allow for movement of the cells themselves, as well as movement of the organelles in the cell. Okay, so again, when you think of cytoskeleton, cyto means cell. Skeleton is just like your skeleton. It's maintaining shape and allowing for movement. Now, as I said, the cytoskeleton has a whole bunch of different protein fibers, and you'll see these written out as actin or microfilaments, intermediate filaments, and microtubules. And all of these filaments are basically involved in helping the cell move and helping organelles move. Actin, you'll see, let me just highlight here, actin is represented here as thin filaments that basically will be involved in muscle contraction. Intermediate filaments, you notice, are now getting a little thicker, and these will help in terms of the nuclear envelope and what's called cell-to-cell junctions, which are basically, think of knitting together or tying together cells. So they help with the attachment to other cells. And then microtubules are the thickest, but they are hollowed out cylinders, and they basically form these tracks in the cells for organelles to kind of move around on. Again, all three are involved in movement in the cell and in maintaining the shape of the cell. In plant cells, you also have what's called centrioles, and the centrioles are basically involved in separating during mitosis. And when you think of mitosis, what that is total cell division. Okay, so when your cells split, you will see the centrioles kind of go to their own side of the cell. So one centriole on one side, one centriole on the other side. Okay, and that way each of the new cells then have these centrioles, and they're believed to then give rise ultimately to what's called cilia and flagella. And as we mentioned in previous slides, flagella help cells move around. They can be those little whip-like structures that propel cells forward. Cilia sometimes look a little bit like shorter version of flagella. They're basically like a whole bunch of little hairs that can also help a cell move around. And so this is to help you visualize what I was just talking about in terms of cilia and in terms of flagella. Again, both of these are those extensions that you'll find outside of the cell, and they basically help with movement. Okay, so cilia will look like a whole bunch of little hairs moving in the wind, and flagella will be the longer extension. And I want to point out that in these examples that I listed here, sperm cell, they're usually using a long flagella, whereas moving mucus is usually using cilia. And that's fun fact, if you've ever heard of whooping cough and you hear someone going constantly coughing, it's because their cilia in their respiratory tract have been destroyed by sometimes, you know, the bacterial toxins, or people can even destroy them through smoking and vaping too much. And so you'll constantly hear the coughing because they no longer have cilia to help move mucus out of their body. And so it does the same thing in terms of individual cells as well. Cilia helps move things. And then we just have this summary chart. Again, when you see summary charts like this, you do not have to memorize this information. It's just something to help you visualize everything in one place. but what's critical is on the individual slides when we went over things like function. Make sure in your study guide for this chapter, you have each of the structures and organelles that we talked about. Make sure you have the functions of each of them, and make sure you emphasize if it's only found in a plant or if it's only found in an animal. And if I didn't emphasize it being only in one or the other, then that means you usually find it in both places. Again, at the end of any chapter, make sure you are updating your study guide that you have to submit by the end of the week, and make sure you're thinking about what multiple choice questions you would like to post on your weekly discussion board post. And when you do your discussion board post, make sure to reply to one other student's post that someone else has not replied to yet. Okay? As always, if you have any questions or concerns, make sure to contact me in the Remind app. It's always better to contact me sooner rather than later. That way I can help you much more as you are studying and preparing and trying to figure out all of this crazy material. Okay? Thank you and have a wonderful day.